and recognise the generous support of our sponsors, Sanofi Genzyme and Genentech, a member of the Roche Group. Thank you for helping to make this webcast possible. The Alliance is an unprecedented global collaboration. It includes 19, that's 19, MS organisations, hundreds of researchers and healthcare professionals, the pharmaceutical industry, companies, foundations, donors, and people who are affected by progressive MS. Essentially, the Alliance has brought the world together to solve progressive MS. And to date, the Alliance has invested over 24 million euros in research, with an overall commitment of 60 million euros through 2025. The Probabilistic Vision Group and Electrical and Computer Engineering Medical Imaging Lab. She's a co-investigator with Professor Arnold on the Collaborative Research Network focused on using artificial intelligence in MRIs. Her research develops new machine learning frameworks in computer vision and in medical imaging, particularly in the areas of neurology and neurosurgery. Professor Robert Fox is a neurologist at the Mellon Center for Multiple Sclerosis and vice chair for research at the Neurological Institute Cleveland Clinic in the United States. He's also a professor at the Cleveland Clinic Lerner College of Medicine. He's a member of the International Progressive MS Alliance Scientific Steering Committee and leads its expert panel focused on fluid biomarkers. Now, Dr. Fox's research includes clinical trials in multiple sclerosis, innovative MRI techniques to ensure to evaluate tissue recovery after injury, and the effects of MS treatments, as well as MS patient decision-making and tolerance to risk. Wow, I mean, I'm in such fantastic company today, just listing all of what you're doing, where you're working, all of your roles. It's amazing. So thank you so much for joining me today. Are we ready to get started? Should we have a live, interactive, brilliant discussion all about biomarkers and what's happening in the area of research for progressive MS? So I'm not going to lie, these have been challenging times for people throughout the world. And it's been particularly hard for people with multiple sclerosis because we've had to deal with chronic disease whilst we're in the midst of a pandemic. So I want to start with a bit of good news from each of our panelists. In terms of progressive MS, what is the one thing that people can be inspired by in terms of treatment and research? Let's begin with you, Professor Arnold. What is inspiring you?
Thanks very much, Tricia. For me, the most inspiring development in recent years has been the development of treatments for progressive MS. As you probably know, we now have ocrelizumab as a treatment for primary progressive MS and siponimod as a treatment for secondary progressive MS. These are major advances in an area where we didn't have treatment before. They're not as good as we'd like them to be. There's a lot of work to be done, but at least they're a start.
Thank you. And Professor Fox, what is inspiring you?
So I think what's inspiring to me is our recognition now of how different progressive MS is from relapsing MS. Okay. And in years past, we've done trial after trial where we've just taken the relapsing MS drug and we've slapped it onto a progressive MS trial in hopes that we would find success. And we did not. We've had failure after failure. And I think we finally learned what we didn't realize, which is that progressive MS is very, very different from relapsing MS. We need to understand better what is driving it, the pathophysiology, or what tissue is falling apart to drive progressive MS. We need to identify better ways of measuring early stage and later stage trials in progressive MS. So I think what's inspiring me is realizing what we really didn't know, which is how different progressive MS is than relapsing MS.
Thank you. And finally, Professor Arbel, so you work in the field of artificial intelligence and computer learning. What inspired you to collaborate with Professor Arnold on progressive MS research?
Thank you very much. So I'll just tell you a little bit about my background so then you'll get a bigger picture. So my area of research is in developing new machine learning methods for a field called computer vision. Computer vision is a field that involves developing computer programs to automatically analyze and understand images. So for example, to get a computer to understand an object in the scene from a camera image. So when I came to the Montreal Neurological Institute to do my postdoctoral fellowship many years ago, I started to realize that machine learning from medical images can lead to significant advances in patient care. And so Dr. Arnold and I have been collaborating for almost 20 years since then. And that collaboration has led to development of sort of new automatic techniques for things like automatic lesion segmentation and detection in MRI acquired during clinical trials for new treatments for MS. And our automatic techniques developed in our lab are used in the development of almost all or most of the new MS treatments worldwide. So when the IPMSA, the International Progressive MS Alliance, developed a program to support new research in progressive MS, the idea is that early prediction of future clinical disability could lead to improvement in patient care and more appropriate treatment selection. And so in progressive MS, there are no MRI markers to reliably predict future disease progression in individuals. So we are working on focusing on developing these sophisticated AI tools for this problem based on MRI, with the goals and hopes of accelerating drug development and predicting patient outcomes on and off treatments from images.
Thank you. You know what? It's so interesting. It's one of these things which for somebody like me who's living with MS, it's potentially mind-blowing. So let's talk about treatment development. We all know that there is an urgent need for new treatments for people with progressive forms of MS. We've already touched on it slightly. There are now so many treatment options for relapsing forms of MS, and it looks like we're going to see even more in the near future. And yet we've only seen limited advances in progressive MS. Professor Arnold, what has been sort of getting in the way of developing these effective treatments for progressive MS?
So Dr. Fox alluded to this. You know, there are challenges to developing effective treatments for progressive MS that are different from those for developing treatments for relapsing MS. And the first one, of course, is that we don't really understand all the mechanisms that are responsible for progression in MS. And so we're not sure what to target with our treatments. If we focus for the purpose of this discussion on inflammation, which is kind of pivotal in MS, we have to recognize that there are two different kinds. You know, there's the kind of inflammation that produces the relapses and the usual MRI lesions, the kind that everybody thinks about. This kind of inflammation influences progression to some extent, but it in fact accounts for only a minor proportion of the progression that occurs. Recent advances in drugs to treat this kind of inflammation, you know, such as the drug I mentioned before, have really been spectacular advances in the treatment of relapsing disease and in the relapsing related inflammation. But they've made only modest contributions to the treatment of progression, you know, supporting the fact that there's something else going on. And that something else is, there's probably multiple other things going on, but one of the most important is a different kind of inflammation, a second kind, which we can call progression-related inflammation as opposed to relapse-related inflammation. And this kind of inflammation is, in fact, resistant to treatment with the drugs that work for relapsing disease. And we need to look for novel treatments that will, you know, target this progression-related kind of inflammation. And these are different drugs, fundamentally different drugs than the ones that have been available for relapsing disease. The development of treatment for relapsing MS has been helped enormously by the use of MRI to measure new lesion formation, either based on gadolinium enhancing lesions or new T2 lesion formation. The MRI is able to show the effects of potential treatments in these drugs and have enabled the drugs to be evaluated relatively early in their development using these MRI biomarkers instead of you know, a clinical outcome such as relapse rate, which require much longer, much bigger, much more expensive trials. You know, the problem is that we have no comparable MRI markers for progression-related inflammation. And so we can't do these relatively small, relatively short, less expensive early trials. And the result of this is that every time a researcher wants to evaluate a new drug for progressive MS, it requires a late-stage trial or two that costs literally hundreds of millions of dollars. You know, an amount of money that is that is a significant barrier even for big pharma companies.
Yep.
And so this is where the IPMSA funded MRI network comes in. We're hoping, as Tal has suggested, that in new kinds of MRI measurements developed with the help of AI, we'll be able to overcome the lack of good biomarkers for progressive MS and make it easier to evaluate new treatments.
Thank you, Professor Arnold. So we've heard you say a number of times the word biomarker. Professor Fox, let's move to you. A biomarker is a critical piece of the puzzle in finding new progressive MS treatments, as you know, Professor Arnold's just spoken about. Can you tell us more about them and how they're used in developing new treatments? I mean, firstly, and I'm sure many people who are watching today are asking themselves is, what even is a biomarker and how are they then used in developing these new treatments?
Sure. So biomarker is a shortened word, or portmanteau, a shortened word for a biological marker, which means a measurable thing or a measurable entity whose presence indicates something, perhaps a normal biologic process, or it might measure pathology, measure what's going wrong in the body. So for example, one can look at the blood count and know how healthy one's bone marrow is in producing the blood cells. You can look at the oxygen level in your blood and know how well your heart and your lungs are getting oxygen into your body and around your body. Those are biomarkers of different biologic processes. So treatment markers or therapeutic biomarkers are a specific type of biomarker. And it's a type of biomarker that indicates a response to a therapeutic intervention or to a treatment. In relapsing MS, as Doug was alluding to, new lesions on the MRI are widely utilized as a biomarker of response to treatments in relapsing MS, that is, to the anti-inflammatory therapies. So if one sees with a new therapy a reduction in new lesions on MRI in relapsing MS, that will have a very tight association with a clinical treatment response, which in relapsing MS would be a reduction in the frequency of the clinical relapses or the relapse rate. So we have a very effective biomarker for relapsing MS. But it raises the question, well, what about progressive MS? And that's where the challenge has been. We do not yet have a sensitive and specific biomarker for use in progressive MS.
Okay, so if I've got this correctly, the bottom line is that having good treatment biomarkers is vital to developing effective new treatments in progressive MS. Have I got that right?
Yes, because it allows us to gain insight into things before we see the clinical manifestation. So for example,
Sure.
A patient might be on a medicine that might irritate the liver. We don't wait until their liver dies to see there's something going on. We measure the liver enzymes in their blood to see, your liver is irritated. We need to take some action. So the liver enzymes in the blood is a biomarker of the underlying process, and it tells us things earlier than we might see clinically. So it gives us greater insight and more sensitive insight into what is going on, whether it's a disease or it's improvement to a therapy.
Fab. So in order to just give a little bit of background to the people who are watching, the Alliance is working on this area in two fronts, really. So firstly, forming an international research network to develop the use of artificial intelligence in MRIs. And secondly, the use of a blood test that could measure something which is called neurofilament light. So we're going to explore each of these, starting with the research in MRIs. Using MRIs in MS isn't new. Most people with MS have had an MRI. Many of us have had them multiple times. I'm often seen falling asleep in my MRI scans. And it's those MRI scans, it's basically in order to know if there's lesions on the brain and or spinal cord and if they're getting better or worse. So Professor Arnold, what's different about what you and your colleagues are trying to accomplish using MRIs?
So we know that MRI lesions associated with relapses are not responsible for the majority of the progression that we see in progressive MS. What we don't know is how to visualize the MRI disease processes that are responsible for the majority of the progression. The usual things that we look at, for example, just looking or eyeballing the MRI, the way radiologists usually do, by measuring things in the lab that we know how to measure, like lesion frequency or changes in brain volume. These are simply not good enough for the predictions that we want to make or for the as biomarkers in the clinical trials that we want to do. And this is where artificial intelligence and more specifically deep learning come in. So we are counting on Tal and her AI and deep learning expertise to figure out what features of the MRI are associated with progression, features that we can't see just by looking at it, so that we can use these biomarkers to facilitate earlier stage clinical trials for progressive MS, and in so doing facilitate drug development for progressive MS.
Okay, so a follow-on question then for Professor Arbel. Could findings from the research network provide other benefits to people with progressive MS besides letting us know if a new treatment works or not? So
I guess the The question is, machine learning or deep learning, these newfangled AI techniques are going to be, we're hoping, helpful in predicting in an individual patient how they will do, will they progress, and the computer is actually doing that internally. It will figure out features for patients with progressive disease, and will internally figure out features that are indicative of future progression. And so, yeah.
Sorry, can I carry on?
So we're very, we have started to have some success in having AI tools predict whether, specific progressive MS patients will progress from their initial MR images when no treatment is provided. So the computer can learn from patient images on placebos, for example, in clinical trials. And then, and we're looking now into sort of being able to predict which individual patients won't progress or will progress on different kinds or classes of treatments. And we're hoping that this will permit clinicians to make informed decisions and offer appropriate drugs and help them with that decision.
That all sounds amazing. It's like it is like I mentioned before. Some of this is mind blowing for somebody who is living with MS. This research that's going on, most of us don't realise the power of combining what is cutting edge technology with scientific research. It has the potential really to answer some of the hardest questions in MS. And also, as you just mentioned, potentially customize treatment for each person. We could have personalized treatments, which would be amazing. So let's now talk about the development of a biomarker that would involve the use of routine blood tests. But it's much more than that. It involves something called neurofilament light. So Professor Fox, you've led an expert panel that the Alliance formed to examine this as a biomarker for progressive MS. Help us to understand what it's all about. So firstly, what is neurofilament light and what is its role in the whole biomarker discussion?
Sure. Well, as you mentioned, the Alliance convened a working group to look at fluid biomarkers, which are biomarkers in the blood and the spinal fluid that might indicate, one, the degree of ongoing injury from MS in the brain and the spinal cord, and hopefully then also indicate a response to therapies in progressive MS. The group looked through the scientific literature to identify different potential candidate biomarkers, and one emerged as looking very attractive, and that is called neurofilaments. Now, neurofilaments are a family of proteins. They are the cytoskeleton within the cells. So they are like the leg bones to the body. They are the steel scaffolding within neurons in the brain and the spinal cord, and they help keep the structure of the cells. So these steel girders help to give the size and the strength and the direction that the neurons and axons are, and they give the stiffness to the cells. Now, when a neuron is injured, these neurofilaments are released. They're released first into the spinal fluid, and then that's the fluid that bathes the brain and the spinal cord. And then these proteins, neurofilaments, eventually move out of the spinal fluid and into the blood. Now, these neurofilaments are released regardless of the cause of injury, released into the spinal fluid and then into the blood. And we can measure them. And fortunately, because they are structural proteins, they're very sturdy. So they don't break down when they get into the spinal fluid, and they don't break down when they get into the blood, because you want something that'll be there for a little while and that you can measure. And it's also specific to the brain and spinal cord and other nerves. So we know it's not measuring liver injury or kidney injury, but nervous system injury. And it's something that we can measure very easily from the blood. Now, one of the drawbacks is that it is not specific just to MS. So a stroke, traumatic brain injury, any injury to the brain, to the spinal cord, to the nerves can cause release of neurofilaments into the spinal fluid and into the blood. But that said, it still is thought to reflect the amount of injury, and therefore has two potential roles we envision. One is to be as a prognostic marker, a prediction of how much injury has happened and how much further clinical decline or disability may be in the future. And also we think it will be a measure of response to therapy. So not just predicting how injured someone is, but also the response to a potential protective therapy or restorative therapy for progressive MS.
Okay, so the Progressive MS Alliance expert panel just published a series of recommendations for basically advancing the use of this neurofilament light as a biomarker in clinical trials for progressive MS. What are the next steps then? How close are we to seeing that happen?
Well, there were a number of recommendations that we made. And one of the key issues is we don't really know how useful this is going to be in progressive MS and progressive MS trials. And so that's what we need to look at. We need to look at neurofilament in previous studies, both natural history studies and clinical trials, to understand what it tells us in relation to the MRI measures of disease injury, as well as the clinical measures. We also need to better standardize how it's measured. There are new laboratory testing that is being developed, and we need to understand how comparable they are from one lab to another, from one diagnostic platform or testing platform to a different testing platform. So those are the sorts of questions that we need to understand more and then apply it to future clinical trials to then correlate it with the clinical disability, because the goal is that this will be useful to measure both the prognosis as well as the response to therapy much quicker with fewer patients, to identify the proof of concept studies that a drug appears to work, so we can then justify doing it in a larger, longer trial that typically costs a lot of money, as Dr. Arnold was mentioning, but to get the proof of concept first to then justify going to the big, long, expensive trial.
Okay, thank you. So we've spoken about the use of this, this fluid biomarker in clinical trials. So how I understand it, the use of neurofilament light could also ultimately be part of patient care. Can you give us an example of what that might look like as somebody who's living with MS, who, you know, goes to my neurologist clinic? How could this biomarker potentially be used as part of my care in MS?
Yeah, well, right now, the main use of a biomarker is in relapsing MS, and that's MRI. We look for new lesions on MRI to identify disease activity even before the patient has a clinical relapse. So what we want to do with progressive MS is to develop a similar test, whether it's by MRI, as Dr. Arnold and Arbel are developing, or by fluid biomarkers, like with perhaps neurofilament, so that in the clinic we can say to a patient, one, you're at higher risk for having more progression, so we need to perhaps be more aggressive with our therapy. And at the same time, we can also measure the impact of the current therapies of, yes, things do look good as measured by these biomarkers, or actually, no, they're not. We need to think about changing therapy into a different direction. So that's what we're hoping to do. In terms of neurofilament, we're not quite there yet, and we need to understand more how to use it in clinical practice. There are some neurologists are using it in relapsing MS as a measure of disease activity or active inflammation. We still don't know how to use it in progressive MS, at least not yet.
Okay, well, even still, I think it's really exciting to see these kinds of advances being made, because they could potentially benefit people with progressive MS. I find this whole topic fascinating, and we could quite literally go on for hours about it. But as we come to our session's close, I'd like to ask each of our guests what the future holds when it comes to developing new treatments for progressive MS. Where do we need to focus next? Professor Arbel, should we start with you?
Sure. So in my mind, artificial intelligence and machine learning really does have enormous potential for assisting in the development of new or better treatments, and also for the ultimate goal, which is precision medicine for people with progressive MS. So I'm interested in focusing new treatment development on particular patient subpopulations within progressive MS. So instead of determining, for example, that if the drug works for most people as is currently done, we could focus machine learning to sort of determine whether it would work for particular patients. So for example, even if there's a failed trial, the treatment could work on a small subpopulation. So if machine learning could actually figure out who that subpopulation is, we could have potentially more treatments available as well. And I guess the long-term goal would be to have machine learning predicting treatment options for progressive MS based on MRI, which is a non-invasive medium, along with potentially any clinical information that is available. So working on them together. So these are some of the directions that we're excited and interested in developing.
Thank you. And Professor Arnold, where do we need to focus next?
Well, I certainly agree with Professor Arbel about the long-term goal of precision medicine. This is where we need to go. But before we can practice precision medicine, we need to have treatments that work, that we can use precision medicine to prescribe. And I think that's what the immediate focus needs to be in the primary progressive or in the progressive MS field. And of course, it's our hope that the AI methods we're working on will be able to contribute to this.
Thank you. And last but not least, Professor Fox.
I think one of the big question marks in progressive MS is what is truly driving it. What cellular process is falling apart or is not working right or is working in the wrong direction that is driving the gradual insidious decline in function? And I think we don't really understand that. And if we understood that better, is it from compartmentalized inflammation? Is it from mitochondria, the energy metabolism part of the cell that is declining? Is it chronically denuded or the lack of the protective sheath on axons? When they chronically don't have that sheath, do they metabolically fall apart? There are a lot of different potential explanations, and we don't really know the underlying one. If we could figure that out with better precision, I think we can target therapies better, and then these biomarkers will be more useful in identifying the potential benefit of these therapies.
Thank you. So in conclusion, well, the International Progressive MS Alliance is basically rallying the global MS community and fueling important research to get effective treatments to people who need them. The Alliance is continuing to inspire hope and also make progress, as we've demonstrated today through our discussions. But we still must do and will do more to end progressive MS. Thank you to our panel of esteemed experts, Professor Arnold, Professor Arbel, and Dr. Fox. And thank you for joining us today. I know that this takes time out of your busy diaries, and we really appreciate it. As someone who's living with MS, it's always such a great opportunity to be able to chat with people who are researching in order to make lives better for us. Please stay connected to the Alliance through the regular webcasts and also your own MS organisation in your various countries. And please share the work of the Alliance with others as well. We need everyone to be part of this effort. Together, we are stronger than progressive MS.